Thank you very much. I want to thank you also, the scientific committee, to give me the opportunity to present here my job. It's a pleasure for me because Madrid is my own city, and I hope you all are enjoying your stay in Madrid. I'm going to talk about exploring new mechanisms to understand the complex B-cell dysregulation present in CVID. So primary immunodeficiency, as you all know, are inherited disorders of the immune system, and they comprise a broad spectrum of disease. Antibody deficiency are the most frequent ones. Common variable immunodeficiency patients suffer from recurrent severe infections, but also some of them present other complications such as lymphoproliferation, gastrointestinal, or also autoimmune complications. Very briefly, laboratory findings include hypogammaglobulinemia, low vaccination response, and also in many patients we found a reduced switch memory B cells. Common variable immunodeficiency disorders is very heterogeneous. There is a high variability in the age of onset since late childhood to adulthood. Also, in some cases, we can see a progression from isolated IgA deficiency to also a loss of the IgM or an IgG levels. So we frequently ask if CVID can only arise as a consequence of sequence variations, because less than 20% of CVID patients present a monogenic defect. CVID is an heterogeneous disease, present a common epidemiology, and also a complex pathogenesis. CVID disorders, we think we need for other approaches. So for all of those circumstances, we decided we have another hypothesis, is that if epigenetic dysregulation might contribute to CVID pathology. Epigenetics refers to those hereditable changes in the chromatin structure that modify gene expression without changes in the DNA sequence. One of the epigenetic modifications is DNA methylation. It's by addition of a methyl group in cytosines at CPG dinucleotides. Dinucleotides, the CPG dinucleotides, tend to be clusters at a CPG island near of gene promoters. So they are interfering with the accessibility and recruitment of transcription factors. Also, the methylation can be induced by proliferation, but also by TET enzymes. So during B-cell differentiation, there are epigenetic mechanisms that control transcriptional programs. So DNA methylome, we know that is modified during B-cell development and affecting several million of CPG sites. So transcription factors, but also epigenetic markers such as DNA methylation, regulates the expression of genes related with B-cell functions. During B-cell development, since bone marrow through the germinal center reaction to finally produce antibody-secreting plasma cells, there is a massive loss of DNA methylation. In particular, here in the germinal center reaction, there is a massive methylome reconfiguration, but also a high proliferation rate. Also, memory B cells are epigenetically prepared to rapidly differentiate into antibody plasma-secreting cells. As you all know, CVID patients frequently present different blocks in B-cell development. So they present an impaired in B-cell differentiation. So we decided to analyze DNA methylation in B-cell subsets. We sort naive and switch memory B cells from CVID patients and healthy donors to analyze the DNA methylation in genes implicated within B-cell differentiation and activation. To that end, our collaborators from a previous work have done a DNA methylome during human B-cell differentiation. So we selected relevant genes, and we selected some CPGs that undergone the methylation from naive to memory B cells. And in fact, what we found, as expected, is that our healthy donors also present the methylation from naive to switch memory B cells. and that CVAD patients, the naive B cells in red, present similar levels from those of demethylation from those of healthy donors. However, we found that there was an impaired demethylation from naive to switch memory B cells in CVAD patients, because as you can see in red, there is a higher methylation levels in the switch memory B cell compartment. But it was only for some of the CPGs, because in others we found normal levels. So that means that it's not a specific phenomenon. However, the intensity of impairment in demethylation was related with B cell phenotype, because for those few patients that have a normal distribution in memory B cell compartment, we found a similar demethylation from naive to switch memory B cells, very similar to those of healthy donors. However, in patients that have a reduction of switch memory B cells, or both unswitched and switched memory B cells, the intensity of impairment in demethylation was associated because they cannot demethylate as healthy donors. The methylation could also be associated with cell divisions, so we decided to study the replication history, the number of cell division in the same samples of healthy donors and CVD patients in all the three populations. What we found it was that from naive B cells from CVD patients, they present more cell divisions than healthy donors, but we don't find any difference in the memory B cell compartment between healthy donors and patients. So we also asked another question: if it is impaired CPG demethylation observed in switch memory B cells from CVD patients could be related to the proliferation rate. We didn't find any correlation in healthy donor, neither for most of the CPG study, but we found a single exception in IDG. We found a negative correlation between higher levels of methylation in switch memory B cells, but also a lower cell division. IED, as you all know, is the master regulator in the germinal center reaction and is implicated in two processes, somatic hypermutation, but also class switch recombination. So we also analyzed the somatic hypermutation levels in the same samples. And we found a severely reduced somatic hypermutation in unswitched and switched memory B cells from CVAD patients. So our study reinforced the hypothesis of altered demethylation during B cell differentiation as a contributing pathogenic mechanisms to the impairment in B cell function and maturation in CVAD. Finally, I want to remember La Doctora Margarita Salas. She died yesterday. She was a referent for the Spanish science, and also she was a pioneer in molecular biology. We thank all the patients and families, all my group, our collaborators, and also the rest of the people that work together with us in the immunology department at La Paz University Hospital. Thank you very much.